Molecular Genetics E. Protein Synthesis, Decoding the Blueprint of Life. Protein synthesis is a fundamental process in molecular biology that plays a crucial role in functioning of living organisms. It involves the creation of proteins, which are essential for various biological processes and functions. During protein synthesis, the genetic information encoded in DNA is transcribed into messenger RNA, mRNA, through a process called transcription. The mRNA then serves as a template for translation, where it is decoded by ribosomes to synthesize proteins. This process occurs in the cytoplasms of cells. What is shown in the image is the process of protein synthesis. The first step is DNA and nucleus serves as a template. The second step is mRNA is processed before leaving the nucleus. The third is when mRNA is formed, it has codons. The fourth is mRNA moves into cytoplasm and becomes associated with ribosomes. The fifth is tRNA with anticodon carries amino acid to mRNA. The sixth is anticodon codon complementary base pairing occurs. 7 is peptide chain is transferred from resident tRNA to incoming tRNA. The 8th is tRNA departs and will soon pick up another amino acid. For the key points, protein synthesis involves transcription and translation. Transcription converts DNA into mRNA. Translation decodes mRNA to synthesize proteins.
Post-translation modification. Fine-tuning protein function. After protein synthesis, many proteins undergo post-translation modifications to achieve their functional form. These modifications can include folding, cleavage of specific regions, addition of chemical groups such as phosphorylation or glycosylation, and assembly into larger protein complexes. So it means atong post-translation modification is pagkahuman sa protein synthesis kasagara sa proteins gaagi sa post-translation modification para makabot sa functional form o mapuslan kani nga modification nga include folding, cleavage, specific regions, addition of groups parehas sa phosphorylation, glycosylation, and mag-assemble into big protein complexes. Post-translation modifications play a crucial role in regulating protein function, stability, localization, and interactions with other molecules. They contribute to the diversity and complexity of the proteome, allowing proteins to perform their specific roles in various cellular processes. Kinahanglan para ma-regulate ang protein function, stability, localization ang interaction sa iyang connection sa uban molecules. Ga-contribute sila sa diversity and complexity of the proteome nga allow sa protein to perform sa ilang tagsa-tagsa nga role sa cellular processes. Gene mutation. Unraveling genetic variations. Gene mutations are alterations in the DNA sequence that can occur spontaneously or be induced by various factors such as radiation, chemicals, or errors during DNA replication. Mutations can have different effects on gene function, ranging from no impact to significant changes in protein structure or function. So it means sa gene mutation is ginalahi niya ang DNA sequence nga gagawas continuously o di kaha gina-induce sa uban factors such as radiation, chemicals, or error during DNA replication. Naay laing-laing epekto sa gene function gikan sa mutation ranging from no effect hangtod naay significant changes into protein structure or function. Classification of mutations includes point mutations, substitutions, insertions, deletions, and chromosomal rearrangements. These mutations can lead to genetic disorders, increased susceptibility diseases, or even beneficial adaptations in certain cases.
What is shown in the image are the different types of point mutation. There is mesence, in which one nucleotide is altered in the strand that change the codon, and also the nucleotide of the complementary strand. This will affect codons, in which will result in the function of incorrect amino acid sequence, which may produce malfunctioning proteins. Next is the frame shift insertion, in which a whole codon is inserted in the insertion site that may disrupt the amino acid sequence. In other words, kaning frame shift insertion kay makaapekto sa sequence sa amino acid nga makaapekto sa paghimo sa protein, like the mesence. And in frame shift deletion, nucleotides is deleted in the structure nga maka-affect yapon sa formation sa amino acids. While in nonsense, the strand stops when a stop codon is met ug diha ra gyud tama ang strand. Point mutation. A point mutation is a change in a single base pair in the DNA sequence, which can occur during DNA replication. This type of mutation can have various effects, from no impact to significant changes in gene expression or protein function. Examples of point mutation include the mutation causing sickle cell disease and mutation in the P35 gene. Overall, point mutation play a crucial role in genetic variation and can have profound effects on gene protein function. So in short, ang point mutation kay occur sa replication. Ug ginipod siya gagay na ay mga varying effects nga significant sa gene expression or protein function. Isa sa mga example niya kay ang mutation nga cause sa sickle cell disease, which is known as sickle cell anemia or anemic, o mutation sa P35 gene or 53 gene rather nga maghatag og stable mutant protein na kung ma-accumulate or matigom kay mahimong hallmark sa cancer.
Original DNA code for amino acid sequence. Ang 3-letter code na CAT nagrepresent sa codons. And ang has na iyang name is which you call an amino acid. Insertion of single nucleotide. As we can see, ang normal unta na sequence is CAT, CAT, and CAT. Ang makita nato na naay letter A na na-insert. So, ang nahitabo, incorrect amino acid sequence which may produce a malfunctioning protein. So, diri na to makita na ang gamay lang nakabag-uhan sa sequence is mag-lead sa dako na kabag-uhan. Ang insertion mutation ga-involve sa addition sa one or more nucleotide sa usa ka segment dito sa DNA, which can disrupt gene function and lead to genetic diseases. For example, cystic fibrosis is caused by an insertion mutation. This mutation plays a significant role in DNA disorder and human health. So, diri na to makita na ang gamay na kabag-uhan is gamatter gyud when it comes to DNA.
Chromosomal mutations. Chromosomal mutations are DNA strand-specific changes that can occur due to irregularities in cell division, crossing over, or fertilization. This can lead to genetic disorders, impacting protein production, developmental difficulties, and health issues, including translocations and deletions. Key points. The key points are gene mutations are changes in the DNA sequence. Mutations can have varying effects on gene function. Classification includes point mutations, insertions, deletions, and chromosomal rearrangements.
Base excision repair corrects small DNA lesions, for example, those caused by oxidation. A DNA glycosylase removes the damaged base short patch or long patch. Repair then fills the gap. Base excision repair or BER corrects small base lesions that do not significantly distort the DNA helix structure. Nucleotide excision repair or NER is the main pathway used by mammals to remove bulky DNA lesions such as those formed by UV light, environmental mutagens, and some cancer chemotherapeutic attacks from DNA. Deficiencies in NER are associated with the extremely skin cancer-prone inherited disorder xeroderma disorder Xeroderma pigmentosum.
DNA mismatch repair or MMR is a system for recognizing and repairing sa mga sayop na insertion, deletion, and misincorporation of bases na mugawas during DNA replication and recombination, as well as repairing some forms of DNA damage. Ang retrotransposons ga-contribute sa structural changes and also, more importantly, sa gene regulation. Retrotransposons are evolutionary widespread genetic elements that replicate pinaagi sa reverse transcription of an RNA copy and integrate the product DNA into the new site in the host genome. Host genome is what you call the affected person.
Kaya nakadevelop og mga repair system sa iyang pag-evolve tungod sa mga factors sama sa exposure sa chemical agents, radiation, ug mga errors during DNA replication. O ato sila nga mga repair system is mentioned in the previous slides, which is base excision repair, nucleotide excision repair, ug mismatch repair. In the shown image, we can see the DNA repair mechanism. Makita nato ang mga damaging agents like DNA replication stresses, oxygen radicals, ionizing radiation, chemotherapeutics, polyaromatic hydrocarbons, UV light, ionizing radiation, o chemotherapeutics. Ang sunod nga makita nato kay ang damaging types sa DNA, which is base mismatches, single-strand DNA breaks, or naay nabalin sa strand, DNA adducts, double-strand DNA breaks. Ug ang sunod nga makita nato kay ang repair mechanism for base mismatches is mismatch repair. For single-strand DNA breaks kay base excision repair. In DNA adducts kay nucleotide excision repair. And sa DNA breaks or dsDNA breaks, which is double-strand DNA breaks, kay DNA break repair or dsDNA break repair. The mentioned repair mechanisms is developed by the cell as it evolves because of certain exposures nga kinahanglan po niya maka-cope up in order to protect the DNA or dili ini ma-damage.
Transposable elements are DNA sequences capable of moving or jumping within the genome. They can change their position and potential influence in gene expression, genetic diversity, and evolution. Transposable elements can be classified into two main types: DNA transposon, which move directly as DNA segments, and retrotransposon, which transpose via an RNA intermediate. Key points: DNA repair mechanism maintains genome integrity. Repair system includes base excision repairs, nucleotide excision repairs, and mismatch repairs. Transposable elements can move within the genome and influence genetic processes.